Welcome to Excellent Grades Academy. This is Dr. Byson EM. Today we're looking at Histology Practical Lab number 8, which is blood. Without wasting time, let's get into this video. So we're going to look at the slides that we're supposed to do in histology. The learning objectives are that the student should be able to describe the component of blood as well as the function of each of the components, to discuss the stages of the development of the cellular elements and give a description of each stage, to describe the structure of erythrocytes and the conditions affecting them, to classify leukocytes and describe the function of each, to describe the light microscopic features of myeloid tissue. Okay, so now the slide that we're going to look at is slide number 10, which is the blood smear, which was stained using gems here. What are we supposed to draw? So first of all, we're supposed to look at erythrocytes. So we're looking at erythrocytes. Okay, so the erythrocytes are just red blood cells. So you select an area where the cells are evenly spaced and you draw 5 to 6 cells. So you draw 5 to 6 cells. Okay. Now observe that they are pink and they are nucleate, which means they do not have a nucleus. They are disc-like structures with pale central regions and a thick, denser outer rim. They can be used as a measure for others formed elements as their diameter is constantly about 5 or 7 micrometers. The second thing we are looking at are leukocytes. Now the leukocytes that we are looking at are about 5 times. So we draw and identify at high power only. You draw the neutrophils. So you draw the neutrophils here. So you draw the neutrophils. So the neutrophils are the majority of the white blood cells, and they are about 70%. So everyone should know that neutrophils are about 70%. They're about 70% of the blood. Okay. They are about 70%. And how do you identify them? The nucleus of neutrophils is multi-lobed, meaning that they have 3 to 5 lobes here. So their cytoplasmic structures are filled with what we call granules. So you draw at least 4 of them at high power only. From neutrophils, you draw eosinophils. The eosinophils have got a bilobed nucleus. So they have a bilobed nucleus, and their cytoplasm are filled with eosinophilic granules, meaning that they are acidophilic. So these eosinophilic granules stain red or orange. So you draw at least 2 of them. The third cell lines for the leukocytes are the basophils. So the basophils as well contain a bilobed nucleus. So they contain a bilobed nucleus and they are basophilic. They stain blue-black. Basophilic means that they are basic. Okay, they are basic. You also observe that you have leukocytes, rather monocytes. So the monocytes, these ones have got a kidney-shaped or bean-shaped nucleus, and they do not have any granules in their cytoplasm. So they attempt to be granulocytes. We say they are granulocytes. They are granulocytes. The first 3 that we looked at were granulocytes, but these ones do not have granules in their cytoplasm, so they attempt to be granulocytes. You draw at least 2 of them at high power only. Then you go to lymphocytes. The characteristic features of lymphocytes is that they have a dark stained and round nucleus, practically filling the whole cell, and they lack granules in the cytoplasm. So the cytoplasm is without granules. So they are also granulocytes. there are granulocytes as well. So you draw 3 to 4 of them. Lastly, on the blood smear, you look at the platelets. Now the platelets, they are blue stained, meaning they are basophilic, they are basic, and they are unnucleated. So they don't have any nucleus. Okay, so you may be able to distinguish the central basophilic zone of the platelet, which is called the chromere or the granulomere, from the peripheral zone, which is light, which is called the hyalomere. So you draw about 3 to 4 of them. So all of these drawings, you are making them at high power. Okay, so now let's look at the slides. So this is the blood smear at low power. So we're only observing at low power. Let's look at this. This is at high power. So this high power representation is showing the erythrocytes, which are red blood cells. So let's pick one. So we said you draw 5 to 6 cells of this. So this here is a red blood cell. You've seen this part here, the round part here, where the nucleus is supposed to be. There is no nucleus, so it is pale. Okay, and the structure of this red blood cell is oval. It's oval. So you draw 1, 2, 3, 4, 5, 6 of them. So you draw them oval. The peripheral ends, these peripheral sides, are darker than the central end, the central portion. So it's enucleate. It doesn't have any nucleus. So this is the blood smear that is showing the erythrocytes. Draw at high power. Draw 5 to 6 cells. Okay. Now, this slide here is the blood smear that is showing the neutrophil. This is the neutrophil here. So as you can see, the neutrophil has got 4 to 5 lobes. So this is the nucleus. The nucleus is multi-lobed. So as you can see, the nucleus has got 1 lobe, the second lobe here, third lobe, and the 4th lobe here. So that's why we say that the neutrophil is bilobed. Why? Because the nucleus has got multiple lobed. It is multi-lobed. The nucleus is multi-lobed, which has 4 to 5 lobes. Okay. So draw this neutrophil at high power, label it, show the multi-lobed nucleus, and label it as a cell. All right, let's move on. Now let's look at the eosinophil. So the eosinophil is this cell here. This is the eosinophil. The eosinophil is bilobed. The nucleus has got 2 lobes. This is lobe number 1 here, and then this is lobe number 2. The eosinophil has got granules. This is a granule, this is a granule here, this is a granule, this is a granule. These granules stain red or orange, meaning that they are acidophilic. Okay. They are acidophilic. So on this structure, you show the bilobed nucleus and you show the red granules that shows the eosinophil. So draw this at high power. Just draw 1 cell and also draw these accompanying red blood cells. So this is a red blood cell. You draw this as well, and you draw this as well. So just draw the eosinophil and the accompanying red blood cells. This is what we call the basophil. So the basophil, we said, is also bilobed because the nucleus has got this lobe and it also has this lobe here. So it's bilobed. Now the difference between the basophil and the eosinophil is that the basophil, the granules here are blue and they are not red or orange, meaning that it is basophilic. Okay, so it stains, the granules inside are blue, they are basophilic. So draw this and label it as this one is a bilobed nucleus and these granules are basophilic. Label it like that. When you draw that, also draw the accompanying red blood cell. This is a red blood cell here, this is a red blood cell here, this is a red blood cell here. So when you draw the basophil, draw 2 to 3 red blood cells as well, just to show that this is a blood smear. Okay. Now let's look at the monocytes. This is a monocyte here. The monocyte has got a bean-shaped nucleus, as you can see. It's like a C. It's a bean-shaped nucleus, or it's a kidney-shaped nucleus. The monocyte is an agranulocyte. 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 Meaning it has no granules inside the cytoplasm. No granules inside of the cytoplasm. Okay. So just draw this, label the kidney-shaped nucleus, and there are no granules. You should note that there are no granules in the cytoplasm. As you draw that, also draw the accompanying red blood cells. These. Okay, since there are two monocytes here, make sure that you also draw this one as well. Okay, let's go to lymphocytes. So this cell here is a lymphocyte. The lymphocyte has got a very dark stained nucleus that is almost filling the entire cytoplasm, as you can see, this one here. And it has no granules, so it's an agranulocyte as well. So draw the lymphocyte with a very big nucleus inside that is almost filling the entire cytoplasm. When you draw that, you also draw the accompanying red blood cells, like this. Okay, now let's go to platelets. Platelets are these cells here. This is a platelet here. This is a platelet here. This is a platelet here. So we said platelets have got two zones. Okay. You've got the zone that is here on the middle that is dark. Okay. That is called the centromere, and you've got the light or the pale stained peripheral part there. That is called the hyalomere. Okay, so just draw the platelets. This is a platelet. Draw the platelet, label the central part, and label the peripheral part as well. When you draw these platelets, you can draw the accompanying red blood cells to show that this is a blood smear. So those are the cells that we are drawing under the blood smear. Draw all those cells at high power, and each cell has to have its own diagram. Okay. So you draw about 8 diagrams. You draw 8 diagrams on the blood smear. The red blood cells, the neutrophils, the eosinophils, the basophils, the platelets, the monocytes, and the lymphocytes. About 8 or 7 to 8 diagrams just on the blood smear. All right, so now let's look at the bone marrow. This is just looking at the development of cells in the bone marrow. So under the bone marrow, what are we going to draw? You examine at medium power and select a suitable area where the cells from the monolayer are. So all cells, especially the erythrocytes, observe them. Identify them at high power. The normoblasts. So the normoblasts are slightly larger erythrocytes with this is the characteristic, with a dark, large nucleus surrounded by a rim of eosinophilic cytoplasm, meaning that the cytoplasm is red. Okay. When the normoblasts exclude their nuclei, they become leukocytes. So you draw at least 3 of them. And then find and draw forms undergoing cell division. So the normoblasts that are undergoing cell division. Secondly, you look at the adipocytes that are also found in the bone marrow. So the adipocytes appear as empty circles and the rich cells population of the hematopoietic cells, meaning that they are a lot. Lastly, you identify the megakaryocytes. So the megakaryocytes are by far the largest cells with a large lobed nucleus resembling the many nuclei stacked together. So you all draw these cells at high power. Let's look at these cells under the histological slides. So this is the erythroblast. The erythroblast is the immature red blood cell. As you can see, it has got a very dark stained nucleus inside this one. Very dark stained, very big nucleus inside that is almost covering the entire cell. So draw 1, 2 erythroblasts. Okay. So this is an erythroblast that has undergone one stage, which is called a normoblast. Okay. So this is a normoblast. A normoblast still has a nucleus. This is a nucleus. And when the normoblast now loses the nucleus, it becomes a reticulocyte. A reticulocyte. Okay, so this is a reticulocyte here. The reticulocyte, where the nucleus was, is empty, so it's pale stained. So it doesn't have any nucleus. So draw the basophilic erythroblast, and then draw the normoblast, and also draw the reticulocyte just on one page. Just label them. Okay. So this is still showing the red blood cells, and the black arrow here, this shows the erythroblast. So already talked about this. It has got a very big nucleus that is almost covering the entire cytoplasm. And this is the polychromatophilic erythroblast. Okay, so it has become a little bit matured. So this is a more immature level compared to this. But you can see that it still has the nucleus inside here. So these are developing red blood cells. You can only find them in the bone marrow where they are made. Okay. So this is the bone marrow showing the neutrophil development. So the neutrophil development here, as you can see, you know that the neutrophil has got a multi-lobed nucleus, but when it is developing, the nucleus is not yet lobed. Okay, so this is the neutrophilic myelocyte, and then this is the early neutrophilic metamyelocyte. Here, the nucleus has begun to at least create some lobes, and then the late neutrophilic metamyelocyte, the nucleus is now separating in two lobes. This is not the mature form yet, but you can see that the nucleus is getting to be multi-lobed. Okay, so this is how the neutrophil develops. First of all, the nucleus is not lobed, and then as it is developing, the nucleus starts to be lobed until it has about 4 to 5 lobes. Okay, so this is the eosinophilic myelocyte as well. This is just shows the neutrophilic myelocytes that are developing. So you start with this one here. So as you can see, this one is a neutrophil, an immature neutrophil, and this immature neutrophil will now go to this stage where it is bilobed, and finally when it is mature, it will go to this stage where it has a lot of lobes like this. Okay, so you can identify that this is a neutrophil and the stage of development by the number of lobes that the nucleus of that neutrophil has. Okay, now this is the last histological diagram. This is called a megakaryocyte. So a megakaryocyte is one of the largest cell in the bone marrow. Okay, this is a megakaryocyte. How do you know? It has a bilobed nucleus. Okay, so this is a bilobed nucleus, and a megakaryocyte is producing these cells here that are called platelets. So this P here just stands for platelets. Platelets. So from megakaryocytes, you've got these segments of cells that are branching off. These are what we call platelets. So a cell in the bone marrow where platelets come from is what we call a megakaryocyte. Megakaryocyte. Okay, megakaryocyte. So draw these megakaryocytes and the branching of these platelets. Okay, so after that histological diagram, now there's this exercise that you need to answer. Okay, so this exercise that you need to answer. So why is the Giemsa stain usually used for blood films? List the components of the extracellular components matrix in the bone marrow. What are myeloid cells? You give examples. Contrast red versus red bone marrow. What is the difference between the bone marrow aspiration and bone marrow biopsy? Which bones are usually used when performing bone marrow aspiration and biopsy? And what is the difference between a thin versus a thick blood film in identification of parasites in malaria? What is reticulocytosis? State any 4 conditions which can give rise to reticulocytosis. And what is the function of the following cells? The neutrophils, the basophils, the monocytes, the T lymphocytes, and the B lymphocytes. Okay, that was the end of the presentations. Thank you very much, and I'll see you in the next video. You can register for lessons with us on 0975497790 on WhatsApp. I'll see you in the next video.